Hello all. In this video, we are going to go over the basics of both RNA and DNA in cells. Now remember, essentially, DNA is the genetic material that is passed on from one cell to another during cell division. Thus, most cells in an organism's body will have exactly the same DNA. However, RNA is usually the expression of those genes, and therefore cells such as liver cells will have different RNA present in them than cells such as skin cells. And so RNA can be ribosomal RNA, transfer RNA, messenger RNA, or in eukaryotic cells, a wide variety of small controlling expression RNAs. Now, once you have DNA, the main gene, it's expressed as RNA, and then it is translated usually into a final protein product. And that final protein product is going to catalyze reactions as enzymes, act as protective agents such as antibodies, function in the complement system. And so each gene typically makes one RNA and typically makes one protein. Of course, with biology, there are lots of exceptions. Generally, adenine hydrogen bonds with thymine with two hydrogen bonds, and guanine hydrogen bonds with cytosine with three hydrogen bonds. And as you can see here, the only way that these four bases can align is that thymine and adenine line up so that the partially positively charged hydrogen is aligned with the negatively charged oxygen, and the partially negatively charged nitrogen is aligned with that partially positively charged hydrogen. And thus they line up that way. And they're always going to have equal distance, and those hydrogen bonds ensure that that alignment is correct. And the same thing is going on with cytosines and guanines. Hydrogen bonding ensures that those are the only two bases that can align correctly like that. If you try to pair T's with C's or T's with G's, the hydrogen bonds do not align. A's and C's, same thing. Hydrogen bonds do not align correctly. And so utilizing the hydrogen bonding, A, hydrogen bonds with T, and C, hydrogen bonds with G. That's how it works in DNA. Now in RNA, instead of thymine, uracil is present. However, the bonding is pretty much the same in that adenine will hydrogen bond with uracil in RNA, just like it hydrogen bonds with thymine, and the hydrogen bonds will be the same. As you can see, the basic structure of DNA bases and RNA bases, you have purines and pyrimidines. Adenine and guanine are purines, a two-ring structure. Cytosine, thymine, and uracil are pyrimidines, a single ring structure. Now, how I traditionally remember the differences between them is adenine and guanines are the bigger molecule, the two-ring structure, the smaller name, and cytosine, thymine, and uracil are the smaller molecule with the larger name, so the pyrimidines. For me, A's and G's are going to be the purines and the two-ring structure. And because cytosine has Y's and T's, I think of cytosine, thymine, and uracil at the end of the alphabet. And so that's kind of how I remember the differences between them, is adenine and guanine, beginning of the alphabet, two-ring structure. Two-ring structure has the smaller name. And then pyrimidines, single ring structure, end of the alphabet, smaller structure, bigger name, pyrimidine. So you should know the basics of which is which and which hydrogen bonds with which. So when we talk about ribonucleic acid and deoxyribonucleic acid, it is important to understand the differences between DNA and RNA. Ribonucleic acid has the sugar ribose and has the bases adenine, purine, cytosine, pyrimidine, guanine, purine, and uracil, pyrimidine. Now, DNA, deoxyribonucleic acid, has the sugar deoxyribose. Now, remember, the prefix DE means no or missing. Oxy is for oxygen, and then ribose sugar. So the sugars are almost identical, except there is a single missing oxygen atom on deoxyribose sugar. Now, the bases of DNA are adenine, purine, cytosine, pyrimidine, guanine, purine, and thymine, pyrimidine. Now, this is where the nomenclature and understanding the basic chemistry comes into key focus. So if you look, you can see the sugars. We have deoxyribose in DNA in blue and ribose in RNA in red. And you can see that the numbers of the carbon atoms are 1', 2', 3', 4', and 5' on both sugars. If you look, you can see on the 2' carbon in DNA, there is a missing oxygen. And in the 2' carbon in RNA, there is an oxygen. So the 2' in RNA, the ribose sugar, has a hydroxyl, and the 2' carbon in deoxyribose does not have that hydroxyl group. Other really important nomenclature. So when we're talking about 3' and 5', all DNA and all RNA start at the 5' carbon. And you can see there's a 5' end, and then there's a 3' end. And the 3' end is that 3' carbon. So the nomenclature of the molecule is very important. All DNA and all RNA only work from the 5' end, the start, and end at the 3' end. So it's the third carbon, and as you can see, both deoxyribose and ribose have a hydroxyl group at the 3' carbon. Now, as you can see in number A, the polynucleotide or nucleic acid, there's a 5' phosphate, and that phosphate group is attached to the 5th carbon. DNA in general is double-stranded, and RNA in general is single-stranded. We talk about viruses, they throw every rule out of the book pretty much, and there are in cases in small cells occasionally that will have double-stranded RNA, and RNA quite regularly in all living cells takes precise three-dimensional shape with areas that are double-stranded. But DNA structure for living cells as hereditary material typically is 2 strands that are anti-parallel. And that the first strand, as you can see here, there's a 5' phosphate at the top of the screen running towards a 3' hydroxyl at the bottom of the screen. Then its next strand, the anti-parallel strand, has a 5' phosphate at the bottom of the screen and a 3' hydroxyl at the top of the screen. And you can see that that DNA is equidistant from each other. Cytosines, guanines, adenines, thymines are holding each other apart and together at the same time with those hydrogen bonds. It's always going to be exactly the same distance, whether you have adenine and thymine hydrogen bonding or cytosine and guanine hydrogen bonding. Those strands are held to a precise distance. And you can see that's what's holding the 2 strands of DNA together. So double-stranded DNA is simply held together by the strength of all the hydrogen bonds of all the bases present. Now in RNA, as you can see, it too has a 5' phosphate and a 3' hydroxyl. You can also see that uracil is present and has replaced thymine. Here's another way to look, and you can see again the backbones of the DNA are going to run anti-parallel. 5' at one end, 3' at the other, and both strands are running 5' to 3'. The strand that starts at the top of the screen and runs to the bottom, the strand that starts at the bottom of the screen with a 5' phosphate and runs to the top with the 3' hydroxyl, both of those are equally important. There is no major strand of DNA, there is no minor strand of DNA. Both strands can have genes on them. Both strands are key. So here you can see, again, the concept of anti-parallel strands. We have 5' phosphates at opposite sides of the strands and 3' hydroxyls at opposite sides. Get familiar with this nomenclature. 5' phosphates, 3' hydroxyl groups, the OH group. Now, in prokaryotic cells, such as bacteria and archaea, DNA typically is a single circular chromosome. Some exceptions do exist. and they do not have organelles inside the cell. And so typically the DNA, the single circular chromosome of prokaryotic cells, exists in the cytoplasm, allowing for quick transcription of the gene into messenger RNA and quick translation of that messenger RNA into a protein product. In contrast, eukaryotic cells with their interior organelles, we usually have the nucleus, which will have linear chromosomes. And depending on the species of eukaryotic cell, you will have one or more chromosomes. Some eukaryotic cells have hundreds of small chromosomes. Other eukaryotic cells just have a few large ones. So it really depends on the species. But they are typically linear chromosomes within a nucleus in a eukaryotic cell. And because of this separation of the nucleus where DNA is, the cytoplasm where most enzymes are, RNA is transcribed in the nucleus and then leaves the nucleus through something called a nuclear pore for translation in the cytoplasm. So for eukaryotic cells, transcription typically occurs in the nucleus, and translation typically occurs in the cytoplasm. Prokaryotic cells, because they lack a nucleus, both transcription and translation occur in the cytoplasm. Again, anti-parallel strands. Be you a eukaryotic cell or a prokaryotic cell, you're going to have 5' phosphates and 3' hydroxyls. The DNA structure is the same. Adenine and thymines, hydrogen bond, cytosines and guanines, hydrogen bond. It's the same for all prokaryotic cells, all eukaryotic cells, and all viruses. Now, when you need to replicate your DNA in order to do cell division, we have DNA replication that is called semi-conservative, in that the original DNA is separated from its double-stranded form into single strands, and those single strands act as templates for newly synthesized DNA. And here you can see the original DNA in blue is being separated, and now we have new DNA in red has been used to, or has been synthesized off the template, which is blue. Because one new strand is generated from one old strand, it's semi-conservative in that one old strand is conserved and one new strand is made. Now, how DNA works is really important with this 5' phosphate. 5' phosphates are energetically unfavorable to add to, whereas a 3' hydroxyl is energetically favorable to add to. So therefore, all DNA and all RNA start at the 5' phosphate, and new bases are added on to the 3' hydroxyl group. It's the only energetically functional way we do this. Enzymes have all evolved to add on to the 3' hydroxyl and not add on to the 5' phosphate. And so here you can see we have a new base coming in. A cytosine is going to come in because the template strand of the DNA has a guanine, and cytosines hydrogen bond with guanines. And so the enzyme, the protein that's going to do this synthesis is usually a polymerase, and polymerases polymerize or generate DNA and RNA. So DNA polymerase is going to bring in the cytosine base and break this covalent bond. Now, this is a covalent bond that, when broken, generates a lot of energy. And so the breaking of that bond releases the energy needed to take that phosphate that's there and attach it to the hydroxyl group that's sticking off the 3' carbon. And as you can see, the 5' phosphate at the other end, there is no hydroxyl to attach to. Making a phosphate-phosphate bond is really hard. That's why we have a special enzyme that makes ATP for us. So it's not going to work unless you have that 3' hydroxyl group. And so enzymes have evolved to take the least energetically objectionable or costly way to do things. And so having a 3' hydroxyl means you can energetically favorably add a base. Now, remember, we have anti-parallel strands, a 5' end and a 3' end for both. And so as you can see here, as you're replicating your DNA, you separate the two strands so both original strands can act as template. And then you have new DNA that's added in. Now, DNA that's added in can only be added to in the 3' direction. So as you can see at the top, we have the template strand running 5' to 3', and it goes that way. And then we have the newly synthesized strand in red going 5' to 3' that way. Then at the bottom, we have the template strand in gray running 3' that way. So 3' is in that direction. And then, as you can see, the direction of synthesis of the newly synthesized DNA in red is running 5' to 3' that way. So as you unwind your DNA, one strand can be made continuously 5' to 3'. And the other strand, as you unwind and open up and separate those strands, is made discontinuously. And so this will show you how it works. As you unwind, you can see that the bottom red arrow, can extend through as you do unwinding and replication. However, the top, as you can see, you've already replicated the exposed DNA. So what happens as you continue going on, you have this unwinding. One strand of newly synthesized DNA is made continuously because it's going in the direction of the unwinding and separating of the strands. And the other strand is always made up discontinuously because it has to be restarted over and over and over again as you unwind more template. And thus, continuous DNA synthesis is called the leading strand because it's running and it's complete. And the other strand that's made discontinuously is the lagging strand. And the lagging strand is made up of something called Okazaki fragments. And the Okazaki fragments are all the pieces of DNA that had to be restarted and restarted and restarted and restarted as that DNA synthesis proceeds. Now, in prokaryotic cells, they have a single circular chromosome typically. So they have an origin where the DNA replication is going to start. And remember, DNA unwinds in both directions. So if you're a prokaryotic cell, you separate the two strands, you start DNA replication. One leading strand goes that way, a lagging strand goes that way. And then on the other part of the chromosome, two templates, right? Double strand DNA gives two templates. Then you are going to have two leading strands and two lagging strands. And as you can see, that will continually replicate through, and you're going to get two new strands of DNA at the end. And for prokaryotic cells, it goes around and around and around. And you can see the origin of replication. You have that newly synthesized DNA bundle opening up. As you can see, the forks are proceeding around the circle, and you eventually get two circular DNA molecules at the end, newly synthesized. And so we have artist renditions at the top, and at the bottom in this gray rectangles are actual scanning electron micrographs of newly synthesized prokaryotic chromosome. You can see it really does look like the drawings. You can see the origin of replication, the replication fork, and how it's proceeding. Now, to start DNA replication, you always need a primer. And this primer is going to denote where to start DNA replication and to bring in DNA polymerase so the DNA polymerase can polymerize or synthesize new DNA. And as you can see here, we have the origins, again, two leading strands. Each strand of DNA acting as a template will have a leading strand, and each strand of DNA acting as a template will also have a lagging strand. So here you can see the two strands of DNA have been separated. You have two origins, two leading strands, and two lagging strands. Now, in eukaryotic chromosomes that are linear, something different happens. Because prokaryotic chromosomes typically are smaller and circular, they only need one origin of replication. However, eukaryotic chromosomes are typically longer and linear, and to speed things up, they'll usually have multiple origin points of replication. so that you have DNA replication bubbles occurring all over the chromosomes all at once, speeding the DNA synthesis. You'll have multiple DNA polymerases polymerizing and synthesizing new DNA based on the template strand. And eventually they'll all come over and line up correctly, and you'll have two complete chromosomes. Now, the enzymes that do this typically are going to be primase, which adds the primer. Now, to start DNA synthesis, primase is going to add an RNA primer. That's just how they evolved. Other names for primase are primosome. And although it's shown here as a green blob, and the green blob looks like one thing, primase is an enzyme made up of multiple subunits. So primase comes in at the origin of replication and adds an RNA primer. And it's kind of like saying, hi, here it is. This is where you want to start replication. Then DNA polymerase, and it's actually DNA polymerase 3 because it was the third DNA polymerase discovered. DNA polymerase comes in, finds that RNA primer, and starts synthesizing new strands of DNA. Again, it's checking to see which bases are present on the template strand and brings in new bases based on the template. So if there's an A there, it brings in a T. If there's a C there, it brings in a G. If there's a G there, it brings in a C. Now, to open up that chromosome, to expose the two template strands, you have an enzyme called helicase. And helicase actually does the untwisting part of DNA replication. Now, because all of those hydrogen bonds want to pull back together, you need a way to hold the strands apart while you wait to get to the replication. And so you have something called SSBPs, or single strand binding proteins. And these hold the two separated strands apart. Now, that untwisting part of helicase's function adds something called torque tension to the rest of the chromosome. Now, to relieve that torque tension, there's another enzyme called gyrase. Now, another name for gyrase is topoisomerase 2. Topo, kind of think about topographic, and then isomers. So basically, topoisomerases change the orientation of molecules. And so gyrase relieves the tension on the DNA that helicase is added by untwisting. And here you can see in this diagram, the primase enzyme that was green last time is now called the primosome. And again, it's simply a different nomenclature. It can vary between book publishers. If you're in Canada or Europe or the United States, sometimes, we typically use the ACE ending, ASE. The suffix means enzyme. And then some publishers prefer the osome, and OSOME means protein complex. And so primosome is just the primase enzyme shown as its multiple subunits. Then we have DNA polymerase complex because DNA polymerase itself is multiple protein subunits. So you separate the strands of DNA. You use helicase to unwind and separate the strands. You use single-stranded binding proteins to hold the two strands apart. You use gyrase to relieve the tension. DNA polymerase is going to polymerize most of the new DNA. DNA ligase is going to join any little Okazaki fragments together. Now, how do you get rid of the RNA primers that primase added? Well, here you can see another little diagram. So we start here at the top with, again, primase adding the RNA primer to bring in DNA polymerase 3. DNA polymerase 3 polymerizes or synthesizes all the new DNA. And then DNA polymerase 3 leaves. Its job is done. Now, another DNA polymerase, this one called DNA polymerase 1, comes in and finds every single RNA primer added by primase and removes it, digs it out, and replaces it with DNA. DNA polymerase 1 is a very special enzyme. and then it's able to both remove bases and replace the bases. Not all polymerases can do this. DNA polymerase is special. And so DNA polymerase 1 removes all its RNA primers, places them with DNA, and then its job is done. And it cannot finalize a final single covalent bond between a 5' phosphate and a 3' hydroxyl. So yet another enzyme called DNA ligase is going to come in and ligate or join all the nicks. So DNA is the genetic material. It is passed on when cells divide. It typically is the same in all cells of a multicellular organism. DNA replication uses parental strands as templates to guide the synthesis of new DNA in semi-conservative replication. DNA polymerase synthesis is always 5' to 3', starting at the 5' phosphate and adding new bases to the 3' hydroxyl. And DNA replication is always going to have a leading strand that is synthesized continuously. That strand is made 5' to 3' off the template strand that is running 3' to 5'. And then it will always have a discontinuous strand that is called the lagging strand, made up of smaller Okazaki fragments that must be restarted because its synthesis is off the strand running 5' to 3'. So DNA guides how cells work. And so DNA can have mistakes or changes, and those changes are mutations. So you always must know old versus new DNA. So if there is a mutation, if there is a change, you can fix it if you catch it. And so usually organisms mark the template strand in some way so that the newly synthesized strand can be checked for mistakes. And here you can see the black dots denote methyl groups. And here you can see the blue strand of DNA and the red strand, double-stranded DNA in a cell, have methyl groups. This is the original strand. So after DNA replication, one strand has methyl groups, marking it as original and template, and the other strand, the newly synthesized strand, lacks that. So the cell can go along and see, is there a mistake? Nope, I don't have to fix anything. Or if one of those G's and C's was switched to an A or a T, there'll be a little bulge because the hydrogen bonds are not correct, or a little divot because the hydrogen bonds are not correct. The cell will focus in on that mismatch, see the original strand is methylated, and it knows to chew out and replace the mistake. So if you don't catch that mistake, mutations will occur in DNA. And that DNA then guides the synthesis of RNA. And RNA guides the synthesis of proteins. So mutations occur in DNA, which will result in errors in the sequence of amino acids that make up proteins. If you have the wrong amino acids present in a protein, you're going to get altered protein structure. And remember, for molecular biology, structure determines function, full stop. So if you have altered protein structure, you will have altered protein function. So E. coli, a very, very common bacteria, can replicate about 1,000 DNA bases per second. So BPS is bases per second. So every single second, a E. coli bacteria can synthesize 1,000 bases of DNA, A's and T's and C's and G's, very, very quickly. And it usually makes less than one mistake per million bases. And E. coli is messy at mutations, and that how it evolves is having mutations. So yes, it does check to make sure that things are right, but it's kind of not great at it, and it kind of wants to make mistakes so that it can evolve and change quickly. And so less than one mistake per million bases is messy for a living cell. So in mammals, let's say we have on average about 3 billion base pairs per cell. On average, we have maybe 3 errors in the entire genome, or one mistake is made per every 10 to the 9th bases that are added. I mean, think about that. I mean, that is just absolutely very, very, very careful in replication. And that's because all of our cells must work together. All of our cells have a very particular job. And a single mistake in one can have drastic consequences for the rest of the body. And we evolve through meiosis and the inner exchange of chromosomes during mating. And so that's how we evolve. We do not want to evolve through E. coli's method, which is making mistakes. So higher eukaryotic organisms have very careful DNA synthesis enzymes and have lots of enzymes dedicated to fixing damage in DNA. Lower eukaryotic organisms like single-celled amoeba also will mate to evolve, but they're usually a little bit sloppier at fixing genetic mistakes. Bacteria want to evolve using mutations, and so they are the sloppiest of the living cells. And then viruses are not living, and they're going to evolve again through mutation. And so they're very sloppy at replicating DNA, especially if they have their own polymerases to make their new genomes. And so as you can see, RNA viruses typically will make one mistake for every single genome, and it will be a tiny genome of just a few thousand bases. Bacteria will have genomes of a couple million bases and maybe will make a mistake. And eukaryotic cells will have billions of base pairs and will probably make less than one mistake. So again, if you're a complex multicellular organism, you do not want mutations. If you are single-celled bacteria, you kind of want some mutations, but not a lot. So again, what happens is DNA is replicated, and while DNA polymerase is really good at checking, it says do these things, hydrogen bond correctly, sometimes there is a mistake. And so here you can see thymine on the original template strand, base pairs with guanine through wobble, there's just a little bit of hydrogen bonding, and you can see there's a bulge. But what happens is that was left, nothing caught it. And so as you go through the next round of replication, there's the original template strand, and that's called the wild type or natural type before the mutation. But then there's that strand that had the mutation, had the incorrect base. And when that one's replicated again, you get a mutation, and now you have the mutant strain. And the wild type strain has the original genetic information. The mutant strain has that change, has that mutation. So you can substitute bases, A's to T's to C's to G's, and you can substitute a single base. And that one A switches to a T, or a G switches to an A, or something like that. Bases can be added in, one base or two bases or 50 bases, or bases can be deleted, one base or two bases or three bases or 50 bases. And that all kind of depends on which polymerase is polymerizing, what region of the DNA is being polymerized. Is it repetitive? Is it not repetitive? So there's a lot of really fiddly little things that feed into what kind of mutation is more likely than others. So we're going to talk about a very common mutation that gives rise to sickle cell anemia. So in amino acids, they're usually going to be hydrophilic, water-loving, or hydrophobic, water-fearing. Now, normal hemoglobin, as you can see, has this series of amino acids, and hemoglobin that causes sickle cell anemia has a different one. And you can see glutamic acid has been switched for valine, and the valine is denoted in red. And what happens is you go from an amino acid that had a very hydrophilic charge to it to something that is nonpolar and something that is completely hydrophobic. And so that is a very, very significant change in those weak forces. Now, when we're talking about red blood cells, the vast majority of their structure is simply hemoglobin for carrying oxygen around in our bodies. There's no nucleus, there's not really anything else in that cell other than hemoglobin, water, and oxygen to move around our body. They can take in carbon dioxide to remove waste, but it's pretty much just moving oxygen around our body, binding it to hemoglobin. So if you disrupt the structure of hemoglobin, you're significantly affecting that red blood cell. So it goes from glutamic acid, a charged amino acid, to valine, an uncharged amino acid. Now, the structure has the same amount of covalent bonds. It's simply you're going from something that is hydrophilic to something that is hydrophobic. Very, very, very different weak charge. Very, very different structure. Very, very different function. The structure is wrong. Therefore, the function is poor. Red blood cells will sickle because you have this hydrophobic residue that doesn't want to be near anything that's wet, and so will sequester itself away from anything that is charged. Now, the sickling means that the red blood cells are not the squishy shape that they should be to get through capillaries and squeeze through various structures in our bodies. We clean them out of our bodies because they are incorrect, leading to anemia, can lead to clotting. It can lead to incredible pain and suffering for someone that has sickle cell anemia. And it's a lifelong disease. And it takes a lot of various medical drugs to try and combat these symptoms. So here we see normal hemoglobin. We have all the hydrophilic residues. They love water. They're fine facing the inside of a red blood cell. And as you can see, each hemoglobin is going to float around in your red blood cell by itself, binding oxygen for us. But in sickle cell anemia, there's that valine. There's that nonpolar residue in red. And so that nonpolar residue doesn't want to be near anything that is hydrophilic, such as the interior of the cell, such as other hemoglobin molecules. It only wants to face something that is hydrophobic. So the valines clump together, and you get these long protein aggregates of hemoglobin all clumped together. You can see that some of the residues of hemoglobin are not mutants. They can still bind oxygen. But that valine one, they're sealing themselves together, hydrophobic to hydrophobic, water-faring to water-faring, clumping up, forming those long fibers. Now, how did we get from there? So here is a codon table, and you can see all the various amino acids. You can see glutamic acid has the codons GAA and GAG. And you can see valine has a wide variety of codons, GUU, GUC, GUA, and GUG. And you can imagine that GUG and GAG, all you have to do is switch A's and U's. And GUA and GAA, all you have to switch is a U to an A. One single DNA base results in a lifetime of pain and this sickle cell anemia. Another change of genes is going to be cystic fibrosis. This is where we have, typically, there are multiple types of genetic cystic fibrosis. The most common one is 3 bases of DNA are deleted. Okay, all that is. So cystic fibrosis is a disease that produces the body. It's going to form in every single mucus layer in our bodies a thick, sticky mucus. Now, normally, we want thin, kind of runny mucus that we're continually making and continually flushing. By flushing this, we ensure that viruses, bacteria, and pollutants are moved through our body and removed. Now, if you have thick, sticky mucus, it does not flush well, and it builds up, trapping pollutants, trapping viruses, trapping bacteria. And so, anywhere you have mucus normally secreted, in cystic fibrosis, you're producing this thick, sticky mucus that does not flow correctly. Now, the sticky mucus results in life-threatening lung infections and serious digestive problems. Our entire digestive tract is coated in mucus. And so the average lifespan of someone with cystic fibrosis up until about 3 or 4 years ago, when a brand new drug was invented, was about 37 years old. And that was, in the UK and Canada, in the United States, with fairly good access to healthcare. Nowadays, with that brand new drug, the lifespan has extended, but it's, again, permanently on this drug. All forms of cystic fibrosis are caused by a mutation in the cystic fibrosis transmembrane conductase regulator, the CFTR gene, which regulates how we make that mucus. controls it so it's nice and runny, so it flows. As I said, the most common one, the most common genetic cause is 3 bases being deleted in the gene of that CFTR. Now, normally, the CFTR transports chloride ions from inside the cell to outside the cell. If it wants to make the mucus a little bit more runny, it ensures that more chloride ions are going out, so it pulls more water out so that mucus can run. But we have mucus in our airways. So if you have cystic fibrosis, you make thick mucus in your lungs, in your trachea. You'll have thick, sticky mucus in your nasal passages. But we have mucus in lots of different places, including our digestive tract. So typically, with that thick, sticky mucus, people with cystic fibrosis are usually malnourished in that they cannot absorb the right nutrients. The pancreas, the liver all have blocked ducts, so they cannot secrete enzymes. The skin is going to need mucus to function. Glands need to secrete various chemicals and molecules. And so you have this really salty sweat. In the reproductive organs, the cystic fibrosis patients are often going to be infertile. Again, there's mucus in there. That thick, sticky mucus prevents normal function. And this CFTR is just amazing. So the most common form of cystic fibrosis is caused by a deletion of the bases CTT, which code for phenylalanine. Okay? As you can see, we have that little triangle, which means delta, and delta means deleted. So here we have in delta deleted, F for phenylalanine, the 508th amino acid. And you can see that's a big protein. It has all those turquoise up and down in the plasma membrane. It has the green spheres, that big red R domain. I mean, there are thousands of amino acids that make up this molecule. You remove one, just one of those thousands of amino acids, you no longer have a functioning pump. And so cystic fibrosis patients have that thick, sticky mucus. There's one particular bacteria called Pseudomonas aeruginosa. And Pseudomonas aeruginosa really loves that thick, sticky mucus. And so cystic fibrosis patients almost always have a permanent lung infection of Pseudomonas aeruginosa. They can sometimes keep it under control with antibiotics, but lots of Pseudomonas aeruginosa bacteria are antibiotic resistant. And thus, maybe a 16-year-old with cystic fibrosis might have the lung function of an 80-year-old, and that is actually fairly typical. Now, where do we have these pumps? Well, it's not like there's just one pump for every cell. No. When you look here, we have some lung cells, and every single green dot there is one of those pumps. You have one large blue nucleus. You have actin making up the cell structure in red. And all those green dots are that pump. And so cells that have that pump are covered with them. And if every single one of those is non-functional, I mean, that's a huge change for that cellular environment. Okay, so we have DNA that is transcribed into RNA, and that RNA is translated into a protein product. So typically we have genes in DNA, and these are the kind of the functional unit of DNA. And a gene is a stretch of DNA, generally anywhere from 2,000 to 50,000 BP or base pairs long. And each gene codes all the information necessary to build one particular protein. And how we code for that is the sequence, A's or T's or C's or G's. In eukaryotic cells, protein synthesis occurs in the cytoplasm, and DNA synthesis and RNA synthesis occurs in the nucleus. And no matter if you're a prokaryotic cell doing both transcription and translation in the cytoplasm, or a eukaryotic cell doing transcription in the nucleus and translation in the cytoplasm, you need a messenger to carry the information from DNA to the thing that's going to make your proteins, the ribosome. And you use messenger RNA to do that. RNA is only made when that cell is ready to make a protein. So If a cell has 30,000 genes, every single cell doesn't need all 30,000 genes all at once. It might need a gene for, let's say, iron consumption for 10 minutes, but then it doesn't need that gene anymore, so it can turn off that gene. So RNA is only made when a cell is ready to have that gene function. That's why I have eyes and a nose and fingers and a liver. My eye cells have all the genes needed to be a liver cell, but it's only expressing eye cells. It's only expressing the genes needed to make a functional eye. My liver, on the other hand, is usually not expressing things like rods and cones, all the genes needed to make a rod or a cone, because it's a liver cell. It has the DNA to make it, but it doesn't need to because it's a liver cell. So each cell in a multicellular organism is going to have a different RNA profile, even though the DNA profile will be the same. So transcription is the process of DNA being transcribed into RNA, and translation is the actual synthesis of that protein off an RNA guide. So you only express genes when they are needed, and cells will take in lots of different signals telling them when to express a gene. These will be extracellular signals from the environment, along with intracellular signals from inside the cell sensing its internal environment. You use RNA, so only the coding information is used. And so typically what you have is chromosomes like this with DNA, made up of DNA with genes scattered around them. Now, these are called intergenic regions between the genes, and intergenic regions are full of information saying, make this gene under these conditions to this amount and turn it off when you get to this. So it has lots and lots of information in the space between the genes. The genes actually carry the information on how to make that protein, but then when to make it, how much to make it, under which conditions to make it, are all that white area, that intergenic region. So it's very important as well. There's lots of extra stuff in DNA. There are ancient viruses, lots and lots of different things. And as you can see, genes are scattered about on the chromosome. So DNA is replicated by DNA polymerases, and RNA polymerases make RNA using DNA as a guide. And therefore, we have anti-parallel strands of DNA. We have something that is called the sense strand, and another name for the sense strand is the coding strand. And that strand will run 5' to 3'. Now, the other strand is known as the antisense strand. The other name for the antisense strand is the template strand. The template strand is going to run 3' to 5'. And the enzyme RNA polymerase uses the template strand or the antisense strand as a guide. Therefore, the RNA transcript will match base for base the sense strand, also known as the coding strand. So if the sense strand has an A, the messenger RNA transcript will also have an A, because RNA polymerase used the antisense strand as a guide. And that antisense strand, called the template strand, had a T. And so RNA polymerase said, oh, there's a T. I need to bring in an A. Both strands of DNA are sense strands, and both strands of DNA are antisense strands. It simply depends on which particular gene you are in the process of transcribing. Here is a map of E. coli's genome, and each little dash is a different gene in that chromosome, and they have incredibly complex genes. Both strands of DNA are template, and both strands of DNA are coding. And you can see here, where there's a blunt end, that's going to be the beginning of a gene going towards the arrow end. And you can see they go both ways. There are arrows that point that way, and there are arrows that point that way. Both strands are coding, both strands are template. It simply depends on the particular gene in question that you're looking at. So this is a tiny part of a gene region. That's actually more than 24,000 bases that you see there. This is just part of one gene on one chromosome. I mean, that's a lot of information. That is a huge amount of energy that would go into replicating that. Now, this is the entire messenger RNA sequence of that very gene. This is not all of the gene. I can't fit it on one slide, and it just turns into blurs if I fit it all on one slide. But this is the entire RNA. You've really slimmed down the amount of information. This is all you need to make this particular protein. And then this is the translated final protein product. All of those, all the codons in there are translated into this small protein product. So it's really amazing that all of that information can be condensed down into this protein product. This protein is actually kind of fun. It's something called a fork-head box protein, and it actually controls the expression of quite a few other genes in neurons. And this actually is part of the reason we have speech, in that it controls how neurons are functioning correctly, which genes they're transcribing, allowing us to speak. One tiny little protein from that tiny little messenger RNA from that big old gene helps us speak. So, in RNA, uracil has replaced thymine. They're very similar structures, and the hydrogen bonding adenine to thymine, adenine to uracil is very, very similar as well. Even though there's that, you can see it circled in red, the CH3 group in thymine, and it's not present in uracil, that's not going to affect the hydrogen bonding. And so adenine and uracil are still going to do that same hydrogen bonding between them. Now, typically RNA is single-stranded instead of double-stranded. An RNA change will be of varying length, from 20 bases of RNA to thousands of bases of RNA in a single strand. And because you can do hydrogen bonding, RNA will fold up into very precise three-dimensional structures. And here you can see A, hydrogen bonding with U, and C, hydrogen bonding with G. Now, one of the main types of RNA is the ribosomal RNA, and ribosomal RNA makes up a huge amount of the ribozyme. And so here you can see the ribosome shown as protein and RNA. All of the ribbons with the sticks out of it are the RNA part of the ribosome, and all of the really curly, ribbony looking things, that's the protein part of the ribosome. And most RNA in a cell is actually making up the ribosome. Now, another form that is very common of RNA is transfer RNA. And transfer RNA, also known as tRNA, transfers amino acids to a growing polypeptide chain. If you flatten out transfer RNAs, you can see they're going to have this typical kind of plus sign structure. They're held together by hydrogen bonds, and you can see C's and G's hydrogen bonding, A's and U's hydrogen bonding. You can see a little amino acid attachment site sticking out, and this anticodon, which will translate. It will hydrogen bond to a codon, thus interpreting the information from the messenger RNA. Now, if you allow them to have their precise three-dimensional form, they're not really plus signs anymore. They're kind of upside down L's, and they do all form that kind of hooked position so that the anticodon is facing down so it can hydrogen bond with a codon very easily, and that amino acid attachment site is again sticking out, and it would have an amino acid added on. Now, the messenger RNA is the last of the three main forms of RNA. And the messenger RNA will encode various codons that were transcribed from the DNA. So the codon would be GCU in RNA, and in the DNA, it would be GCT. Codon 2, ACC in the RNA, codon 2 in the DNA, ACG, and so on and so forth. Because remember, messenger RNA matches the coding strand of DNA. And so the messenger RNA has all the information on how to make that protein. Now, the three main kinds of RNA are ribosomal, messenger, and transfer. But In eukaryotic cells, there are quite a few other kinds of active RNAs. And even in prokaryotic cells, there are several types of RNA that have very important functions. We'll occasionally talk about some of these special RNAs. If you took genetics, you'll probably get a really in-depth look at these various RNAs because they typically control gene expression in eukaryotic cells and defense in prokaryotic cells. You can see there are quite a few different kinds. So I just want you guys to be aware that it's not just messenger transfer and ribosomal RNA. There are other RNAs out there. Each one has a really specific kind of fiddly little job. So just be aware of that. So we have the template strand that RNA polymerase uses to polymerize and transcribe messenger RNA from. The messenger RNA will match the coding strand, T for U, but G for G, C for C, and A for A. And that coding strand matches the messenger RNA transcript. And the messenger RNA transcript is full of codons, and transfer RNA uses its anticodons, hydrogen bonding to codons, to translate the messenger RNA transcript from RNA into amino acids. And I always remember it is transcription is the process from going from deoxyribonucleic acid to ribonucleic acid. From one form of nucleic acid to another, you've transcribed it. But then going from RNA, a nucleic acid, to proteins, amino acids, you're translating. You're going from a nucleic acid to an amino acid. So those codons, 3 bases of DNA or RNA, are how we code for the various amino acids. And so it's the sequence. Is it A, T, C, or G? Is it A, T, C, or G? And the only way to code for all of the amino acids is to use 3 bases in a row. Now, there are 4 potential bases, right? In RNA, uracil, adenine, cytosine, guanine. 4 potential bases in 3 slots. So you have 4 possible possible nucleic acids at 3 positions, gives us 64 codons. You can't use only 2 bases because 4 bases in 2 potential positions gives you 16 codons, and we have 20 or more amino acids. So the only way we can code for all 20 amino acids is if we use 3 bases in a row. So the codon UUU codes for phenylalanine, the codon AAA codes for lysine, the codon CCC codes for proline. And so when we look at tables like this, you can see that like glycine has quite a few codons that code for it. Leucine, I mean, look at leucine, 6 codons that code for leucine. Methionine only has 1. Some of them have more than others. It's kind of, sometimes it is how often they're used, but not always. Like, for example, methionine is the beginning of every protein ever, so it's really important, and that could be why we only have 1. What's really amazing is everything uses this codon. I mean, there's nothing that we've ever discovered that doesn't use this codon chart. Sometimes some cells will replace some of the yellow codons, which are stop codons that stop protein synthesis, but nothing messes with this codon table. The weirdest virus that's been frozen in ice for the longest time, the oldest bacteria from millions of years ago, the weirdest cell under the most extreme conditions, they do not change this table. I mean, this is absolutely full stop. All life, all viruses use this. It's really incredible that no matter how many mutations you go through, this is constant. So we have the codons in messenger RNA, and on transfer RNAs, we have anticodons. So the functions of transfer RNAs are to carry an amino acid, to associate and interact with the anticodon on the transfer RNA and the codon on the messenger RNA, and carry the right amino acid to the ribosome, which is going to do the process of linking those amino acids together. So messenger RNA has the coding sequence and guides the synthesis of a polypeptide chain. Each group of 3 adjacent bases makes up one codon on the messenger RNA. The messenger RNA is translated codon by codon using the anticodons of transfer RNA molecules over and over and over again. And each transfer RNA molecule brings in the amino acid. So you can see the first codon, AUG, on the messenger RNA is translated by that transfer RNA having its anticodon A to U, U to A, G to C. UAC, the anticodon, is the anticodon for AUG, hydrogen bonding. And so on and so forth, all the way from the codon GAA to the anticodon CUU, bringing in a glutamic acid at the end of that transcript. So to join our polypeptides together, you're going to make various amino acids covalently bond with one another, and that will take ATP. And so to add on, like the transfer RNA needs to have its amino acid added before it brings it over to the ribosome. So what you have is an amino acid with its side chain, and that's shown as the R group. And then you prime that amino acid utilizing a unit of cellular energy. And so you have the middle amino acid there. It has the side chain as the R group, and then it has that little AMP accessory molecule added. And that amino acid has been primed for pickup by a transfer RNA. And then that primed amino acid is added onto that transfer RNA. Then and only then can that transfer RNA take that amino acid to the ribosome. And so each particular amino acid has its own special enzyme that does this interaction. So here we have a purple blob enzyme, and it is specific for the amino acid alanine. And it will always be priming the amino acid alanine. And here you can see a cellular unit of energy, ATP, and the amino acid alanine, denoted as a red sphere, enter the purple blob enzyme. And that alanine residue is primed. And then an alanine-specific transfer RNA comes in. And while it is not shown in this diagram, down at that bottom where there's the anticodon, there's a region of that enzyme that's going to ensure that the anticodon is the right transfer RNA. And so here you can see the anticodon here. So here we have isoleucine's specific enzyme shown in light blue, and isoleucine has been primed and is now being added on to the isoleucine specific transfer RNAs. And you can see the isoleucine UAG specific anticodon of the transfer RNA nestling into that specific region on the enzyme. And that enzyme says, oh, you are the right transfer RNA. I will add isoleucine onto you. Or the pink one that's specific for valine. Oh, you have a CAC anticodon. You are the correct transfer RNA. I will add a valine residue to you. And so every single enzyme has that little specific area that's ensuring that the right anticodon is entering, and thus the right transfer RNA has the right amino acid added to it. And then the ribosome is going to do much of the rest of the work. Now, prokaryotic ribosomes are smaller and are called 70S ribosomes, and eukaryotic ribosomes are larger and are called 80S ribosomes. And the S simply refers to when they were discovered. It was a unit of kind of density smears as they were coming out. So here we have a typical ribosome. As you can see, there's a smaller light subunit, or light green subunit, and a larger, slightly darker green subunit. And so the small subunit is made up of various proteins and a single rRNA product, so rRNA, ribosomal RNA. And then the large subunit typically has 3 or more ribosomal RNA molecules and also several different proteins, making our functional ribosome. So prokaryotic ribosomes are made up of a large 50S subunit and a smaller 30S subunit. And you can see for the prokaryotic large subunit, it has 2 RNA molecules. And the small subunit has the 16S ribosomal RNA molecule, and that will become very important later on. So the 16S ribosomal RNA, very important for bacteria. And then the eukaryotic one has a large 60S subunit, and as you can see, multiple RNAs, and then a smaller 40S subunit. Okay, but as you can see, the structure is basically the same. The large subunit has 3 large holes, an A, a P, and an E site, and the small subunit fits in there, forming the bottom part of the A, P, and E sites. A is the amino acyl site, P is the peptidyl site, and E is the exit site. So A is where transfer RNAs bring in new amino acids. And so I always think of A as the acceptor site because new transfer RNAs are being accepted there. The peptidyl site is where the peptide bonds are formed. So the P site makes sense. And then E for exit. Very, very simple. So when you're looking at the ribosome, this is actually how they set up. The A site is where the new transfer RNAs are coming in. The P site is where the current codon is being translated and the polypeptide product is going to stick out directly from that P site. And then used transfer RNAs will be exiting from the E site. Now, if you are a bacteria or a eukaryotic cell, you will often have multiple ribosomes translating the same RNA product at the same time. But if you are a bacteria or archaea, a prokaryotic cell, you can do something special because you do not have a nucleus. Remember, prokaryotic cells have their typically single circular chromosome in the cytoplasm. So if they need to make a gene very quickly, they can be in the process of transcription and be actively transcribing a gene, while at the same time, ribosomes are actively translating that very same transcript. The transcript has not been completed yet, and yet the cell is already translating. And it does this for lots of different genes. This is not just some kind of critical emergency thing. It's done for lots of different genes. And so by transcribing and translating at the same time, bacteria are actually able to make proteins within a few seconds of the stimulus. And so here you can see a diagram of it. Their RNA polymerase is polymerizing a RNA transcript off the DNA. You can see the 5' end of that transcript is all the way at the bottom. Ribosomes are attaching and translating that protein product. And you can see up at the top near the 3' end of the actively transcribed transcript, you have the growing polypeptides that are getting bigger and bigger and bigger as those ribosomes move along the transcript. Eukaryotic cells, because transcription occurs in the nucleus and translation occurs in the cytoplasm, cannot do this. So when you are translating, here we have in the P site the beginning transfer RNAs, and you can see AUG, the start codon, with methionine, the first amino acid of every protein ever discovered. And then the next transfer RNAs coming in. In the messenger RNA in the A site, the codon is CCG. And therefore, the transfer RNA coming in is GGC. And that codon, CCG, codes for the amino acid proline. And so the transfer RNA heads towards the A site. It interacts and hydrogen bonds between the codon and anticodon. And then the methionine is removed from its original transfer RNA and added to the proline of the incoming transfer RNA. And now you can see why they're that upside down L shape. So that where the amino acid sticks on is sticking from the A site into the P site. And so you just keep adding on to that amino acid. Once it has been, the methionine or the amino acid has been removed from the central transfer RNA, that used up transfer RNA moves to the E site and exits. Then the whole ribosome moves down the transcript. And you repeat the process over and over and over again. The ribosome moves down, moving the codon that had been in the E site. It's now gone. We move the codon that had been the A site to the P site, opening up a new codon in the new A site, and so on and so forth. And it reads down from the 5' end of the transcript all the way down to the 3' end of the transcript. Amino acids are always removed from the central transfer RNA onto the A transfer RNA. So they just keep moving them over, moving them over, moving them over, moving them over, growing that that polypeptide, and then allowing that used up transfer RNA to exit and be reloaded with a new amino acid. And eventually you come to the end of your codons. You come to the end of the transcript, and then you're going to use a stop codon. And that stop codon tells the ribosome, hey, you can be done. We've reached the end of what we expect to translate. And so that stop codon brings in something that's not a transfer RNA. Instead, it's something called a release factor. And that release factor decouples everything. It removes the small subunit, the large subunit, the messenger RNA, and that protein product. And all of those things can then be reused. The small subunit can go find a new messenger RNA, the large subunit can go find a new small subunit, and the release factor can go find the new stop codon. So all of those are reusable, and now you've translated the protein product. So remember, in prokaryotic cells, you can be actively transcribing genes off the double-stranded DNA using RNA polymerase. And as that RNA polymerase is polymerizing the transcript, you can have ribosomes translating that transcript to a polypeptide protein product. Okay? Now, both eukaryotic cells and prokaryotic cells can have multiple ribosomes on the same transcript. If you need a lot of that protein fast, that's the easiest way to do it. So multiple ribosomes on the same transcript are called polysomes. And polysomes exist, as I said, in both eukaryotic cells and prokaryotic cells. And so here's another way of looking at it. And you can actually see from the artist's rendition here in green and brown, and then the actual microscope image over here in the grayscale, you have one messenger RNA transcript, lots and lots of ribosomes attached with lots of growing polypeptides. And you get lots of proteins very quickly. So if you need a protein as quickly as possible in something like an emergency, you would have polysomes, multiple ribosomes on the same messenger RNA transcript. After translation, there are lots of post-translational processes that can occur with proteins. They can be chopped up into smaller functional units. They can have sugars or phosphates added. They can have various ions added. There are lots of things downstream that we'll discuss when we talk about proteins. Okay, that is it for this lecture, and I hope you all have a good day.